So what I want to do is give an overview lecture on detecting selection in general, and then we've got a bunch of lectures that then drill down in detail. So I'll be giving examples here I'm going to go back and look at in detail. So today's is largely kind of an overview, because once you get down the weeds, it gets kind of weedy. So I want you to keep the big picture in mind, right? Likewise, if you read chapters 9 or 10 in the book, the way I've structured them is the first part, is I think clearly laid out, gives you kind of an overview of all the methods. Then each of the methods is examined in kind of gory detail. So what I'm going to do is kind of give you that overview of the different methods to kind of let you see the big picture, and that's the most important thing. The details are stuff you work out once you know the big picture, but the big picture is what to keep in mind. So here's a big picture lecture. So again, the issue is I simply have marker data. I either have a candidate gene or a set of candidate genes, or I'm simply scoring a random genome. And I'm trying to find some signatures of selection in that genome. Okay? The bottom line is I'm looking for loci showing departures from the neutral equilibrium model. And again, as I stressed several times, rejection of this model could mean I've got non-neutral alleles, or it could mean I'm not at equilibrium. And that's a very important distinction, and most tests struggle with that. I want to talk about what types of selection are of interest. I want to talk about time scales, because the time scales give you different tests and give you different signals. The key points are false positives are very common. Not all selective events will be detective, and those that are likely represent a biased sample. These are the key points to take away. Negative selection is common, right? Mutations arise which are deleterious, they get removed. This is simply background selection. We can call that negative or purifying selection. This leaves a strong signal throughout the genome. Number one, we see faster substitution rates at silent sites than replacement sites. A replacement site is a site where the codon change results in a change in the amino acid. We expect most of those to be deleterious. A silent site means a codon change doesn't change the amino acid. That could have an impact on codon usage bias or other things. We don't expect that to be nearly as dramatic as changing an amino acid. Comparative genomics. So why do we care about the dog genome sequence or the mouse genome sequence? What you can do is take the dog or mouse genome sequence and line it up with our sequence and look for regions which are conserved. Obviously, genes are conserved, no big deal, but we see conserved non-coding regions. A conserved non-coding region means that region, for whatever reason, is under selection. Otherwise, the sequence would diverge. So the fact that we've got purifying selection means that regions of the genome which are conserved for long periods of time are under selection. And a lot of those turn out to be in non-coding regions, and we're not quite sure what they do. They're probably involved in regulation at some level. So when I talk about the search for selection, I'm talking about the search for selection other than negative. Now this is quite interesting because we can use other genomes to ask about sites outside of coding regions, which may be quite important. So that's non-trivial, but evolutionists are generally interested in searching for selection that's signatures of adaptive, positive changes, alleles arising which are favorable and fixed. So positive selection, an allele is increasing in frequency due to selection. It can either be a new mutation or a previous mutation that was neutral or even slightly deleterious, whose fitness has now changed as the environment has changed. We would call these things adaptive changes. I have a mutation that's now is now favored, and it's being increased in the population. Balancing selection is when alternative alleles are favored by selection when rare. So examples of that are overdominance, sickle cell anemia. If the sickle cell allele is rare, there's a tendency to increase its frequency. If it's sufficiently common, there's a tendency to decrease its frequency. So these balancing selection models are People think about them a lot, but there are very few examples. Warfarin resistance, some thalassemias, sickle cell anemias are examples where the heterozygote had the highest fitness. That was thought in the 50s to be an important model for how mutation is maintained, but we don't see a lot of evidence for that. It may be that it's hard to detect, but we don't see a lot of true heterozygote advantage. Examples, though, are the major histocompatibility system, and the example there is probably frequency-dependent selection. Another example is self-incompatibility systems in plants. In both those cases, if you're a rare allele, you have an advantage. In a self-incompatibility system, it means you're almost never landing on a plant that has your type, therefore you pollinate. In MHC, it means you have a rare MHC. It's not likely to be a target for a parasite or a pathogen. So MHC is well known to be under balancing selection. That's not the same as overdominance. Balancing selection simply means, for whatever reason, when an allele is rare, it has an advantage that pushes it into the population. So the search for selection, then, is the search for signatures of either positive or balancing selection. Positive means they sweep through, or at least sweep through before the environment changes, and balancing means that they're basically, selection is preventing them from being lost when they're rare. So a key issue is time scale. We have kind of two different time scales we can think about. The ecological time scale, and again, I'm not going to put numbers on these, the rough feeling. It means an allele is either currently undergoing selection or has very recently undergone selection, either to fixation or to some intermediate frequency before the environment changed. We can then use a population sample to detect those. For example, patterns and level of polymorphism. The key feature about these ecological events is that a single event can leave a signature. So for example, Biston bechellaria, its melanic form, if you look around that as it's sweeping through the population, there'll be a signal around that gene of interest. On the evolutionary time scale, a gene or a codon experiences repeated adaptation events, either within a phylogeny or over time, over a long period of time. So this requires polymorphism data, so ongoing or just completed single events. This requires between species divergence data. This only informs us as to a long-term pattern of selection over a gene. So we can detect many, but not all, single ongoing events if they're large enough. If I have a history of repeated selection, I can occasionally see signatures in that gene. So for example, let's take alcohol dehydrogenase. I might take a signature right now that one site is under strong selection being swept through. If I then look 10 million years from now, that swept through gives me an additional substitution. One additional substitution isn't much in the background of random substitutions, so that big event that I would catch in ecological time wouldn't leave a signal in evolutionary time. I need multiple adaptive events in a gene to see that signal. In the extreme, multiple events within a codon, and only certain types of things do that, like arms race genes. Like, for example, a pathogen avoidance. So I have a certain sequence, a pathogen adapts to the target gene, I readapt, they adapt, I readapt. You have this arms race going on at a small number of genes, where over time you'd see this cumulative signal arise. So you have these two time scales: ecological, ongoing, or very recent events using polymorphism data; evolutionarily, multiple repeated events either over a gene in a species over time, or over a codon or a small region in a species phylogeny over time. And these require multiple events: polymorphism data, single events, short time, long time, multiple events, divergence data. So you have these two different classes of tests. These are discussed in Chapter 9. These are discussed in Chapter 10. So the classic tests in here, there's really only four. There's McDonald-Kreitman, Hudson-Kreitman-Aguade, HKA test. Those look at the polymorphism to divergence ratios. Then there's the Ka to Ks ratio. is there an excess rate of adaptive versus synonymous substitutions? Then there are likelihood codon models. Those are basically the 4 types down here. In here, there's literally 100 different tests that we'll talk about, not in detail. We'll give the overview of those. So we have these 2 different time scales. Short time scale, single event polymorphism data, long time scale, multiple event divergence data. So tests for selection using sequence data have this broad distinction of different classes they fall into. So questions about that? Again, this is an overview. We're going to go and talk. I think we got 4 lectures in here and 2 lectures in there. So you have plenty more lectures left covering these things. So let's look at some basic approaches. We'll talk about a lot. This is not all. This is kind of to get you started thinking about it. So an obvious approach to look for selection is you have a population that's sampled at 2 or more time points, or a population, 2 or more populations that are sampled. You can look at allele frequency change over time or allele frequency divergence over samples. So both those, if the allele frequency change is excessive relative to drift or not enough relative to drift. Remember, drift says you shouldn't stay static, you should move. We can then detect selection on an ecological time scale. So these require special data. They require basically data from 2 time points. The longer the time point, the more the power, because with weak selection, just 2 generations isn't enough. Multiple population samples, so these are things like McDonald-Kreitman test, these are things like the Fisher-Ford test. You can look at excessive LD, polymorphism data. There's a huge number of tests here. Then you can look at allele frequency and site frequency spectrum data. So again, these require down here, these are polymorphism data. These are data from different populations, either the same population at different time points or different populations at the same time point. So you have different types of sample requirements. You can look at methods for, these are ongoing or recent selection. By recent, I mean, within NE generations ago, just completed. You can then look at methods for detecting repeated selection over multiple sites in the same gene. The classic is look at the ratio of polymorphism to divergent at silent sites and replacement sites, and those are called McDonald-Kreitman and HKA tests, and that acts somewhere on the ecological evolutionary time scale. Excess polymorphism is ecological scale, excess divergence is biological scale. Then you can look at a very special class of events, and these are where you get repeated positive selection over a single site. So I have a phylogeny, and let's imagine that phylogeny, example I'll give is I have several species of Arabis. Arabis is a mustard, a crucifer, Brassicaceae, related to Arabidopsis. It's got fungal pathogens that attack it. The plant counters those fungal pathogens with chitinases, right? Chitin is what fungi have and insects have. Chitinases can attack certain fungi. The fungi then adapt their chitin structure, be resistant to chitinase, and the cycle continues. If you look over a phylogeny of the Arabis, what you'll see is excess number of substitutions in the chitinase genes over that phylogeny. So it's a very special event, an arms race gene. But those things where you have selection that generates repeated positive selection at a single site over multiple species, they're tests to detect that. If you think about this, you can think of lots of examples that don't fall into these categories, and therefore you won't detect them. And this gets back to this main theme of you're unlikely to detect most types of selection that have occurred. Number one, if they're not occurring now, these tests won't pick them up. Even if they are occurring now, these tests only pick up a fraction. Number two, if they haven't been multiple enough or enough at the same site in the past, you're not going to pick up that signal of a single gene. You'll have to have some way of amalgamating signals over the whole genome. So this is kind of an overview of kind of where we're going. This is the stuff in chapter 9. This is the stuff in chapter 10. So this kind of repeats what I said before. We have bias scans for selection. So currently or very recent selection at a single site requires a rather strong selection to leave that signal. A soft sweep or a weak partial sweep or a polygenic isn't going to give much of a signal. So small shifts in allele frequencies in multiple sites won't leave much. We've got a time window to detect these events by many of the methods. Recurrent selection requires multiple substitutions over a known phylogeny in the same codon. This is okay for arms race genes where I have a competitor, I do something, they counter, they counter, I counter. Those often results in independent events at the same codon over phylogeny changing. But these are likely not typical. So a bunch of things can fall out, and that's why I say test for selection taken as a whole group, select a biased set, and therefore looking at positive results in these tests and trying to infer about selection in general is very misleading because these tests don't randomly detect all types of selection. They detect specific types, and therefore they create a bias when you use those. I could look at recurrent selection at sites over a gene. So basically here is I have a codon or a couple of codons, and I look in a phylogeny at that codon, and I ask, is that codon changing in multiple species? Maybe not the same thing, but is it changing in multiple species? This is slightly different. This is saying I have a gene. I've got two species. I look at the gene in species 1, the gene in species 2. Do I see an excess of substitutions across that gene between the species? So I look at fixed differences between species, and I usually compare that with a level of polymorphism. That adjusts for differences in mutation rate. Those are the HKA and McDonald-Kreitman tests we'll talk a lot about. This requires multiple substitutions throughout the gene for any signal. This will drive me crazy here. And hence, a few critical adaptive substitutions can occur in a gene and not give enough signal. Let me give you a hypothetical example. Suppose that language, the ability to speak and comprehend, is a single mutation. When it arose, it gave a big signal. That signal's gone, it's in the past. Now I look at those genes, that gene that's involved in that. There's a bunch of substitutions that have occurred by chance. I have one extra substitution that's driven by selection. I'm not going to pick that up. The signal's too weak. So I might expect to see 4 or 5 random fixations. I now see 6. That's not enough. I can't pick it up. So a single really important event in the past won't leave multiple substitutions, and you won't pick it up. Again, coming back to this concept of a lot of events are going to be missed. So power in these recurrent things depends upon the number of adaptive substitutions being significantly more than the background level of neutral substitutions. So here's kind of a cartoon of these ideas. Sorry about that. Yeah. They keep on saying if you bend it right, it stays in. So I have a sample of several individuals from the same population. So here we are. Here is a polymorphic sample. And what I see here is I see this site here. That's the favorite site. I'd see potentially some signals in polymorphism, site frequency spectrum, allele frequency spectrum. One of the tests, LD, might give me a signal. So I detect ongoing or very recent selection. Not sure the N went. Requires a population sample. Look for inconsistencies of the variation with respect to neutral equilibrium model. Again, the inconsistencies could arise because the equilibrium assumption fails. I can detect ongoing selection in a single region because it influences the pattern variation at linked sites. So a single recent event I can detect with a polymorphic sample. Here is that divergence data over phylogeny. So now these are codons. What you see here, I have 3 substitutions occurring. Because I know the phylogeny, I know these are separate independent events. I know these are different events. The phylogeny is really critical to count up the number of events that have occurred. So in this case, a comparison of sequences over a group of species is done on a codon by codon basis and looking for codons with a higher replacement rate than the site average. This requires multiple substitutions in the same codon over a tree. That's not unlikely for certain things. These may all be a site in the chitinase that's independently mutated to give you an advantage to fungal systems, but it requires these multiple substitutions. It requires special types of selection to generate these patterns. Finally, species 1, species 2, a pattern of fixed differences between species. So I basically sample species 1, sample species 2, get a polymorphism sample, and I compare the divergence data with the polymorphism sample, and that basically requires an excess of divergence over the background level expected from polymorphism, that is, from neutral fixations. So I require a lot. And as we'll see, one of the cool things about these methods is we can actually use them to estimate what fraction of substitutions are adaptive. That's kind of cool. Yeah. Yeah. Good question. So what we're looking at here is QST is focusing on you give me a trait, and for that trait, I compute variances for that. Here, all I simply have is a DNA sequence. I don't know what it does, and I'm simply asking if I look at two species and I see a certain amount of divergence, is that pattern divergence consistent with what I see from the polymorphism data? We'll give an example in a second. It's called McDonald-Kreitman test. And so in that case, I'm simply using sequence information, nothing at all about traits. So FST has to do with how allele frequencies are diverged in space. QST has to do with how genetic variances in space. This is simply, I do the following. I get a sequence from species 1. I get a population sequence from species 2. I use species 2 to estimate polymorphisms. I look at species 1 versus species 2 to estimate divergence. That's kind of the way these methods work. And again, when we go through them, I think they'll make a bit more sense. I'm giving you the overview, big picture lecture here. We'll have 2 lectures on McDonald-Kreitman. We'll drill down in that. So I guess when we look at your question is, suppose it's the case you have this phylogeny and you have a star here and you've got 8 species over here and 1 species over there. So maybe that mutation then caused this to blow up. So you could certainly look at things like that. The problem is, you know, there are lots of different sites you could look that would probably show a similar pattern. Because all you need to do is if you've got an unbalanced phylogeny, any mutation that occurred down here is going to give you that signal. So basically you're inferring the mutation caused the unbalanced phylogeny, rather than the fact that if you have an unbiased phylogeny, any mutations which appear early would seem to be correlated with that unbalanced phylogeny. So it's hard to deconstruct that. People can try and make very clever arguments, but again, you know, you have to remember science and religion. In religion, the burden of proof is on the non-believer. In science, the burden of proof is on the believer. So if you want to make something, it just can't sound plausible. We can all make wonderful stories that sound plausible. You've actually got to go in and be much more rigorous than that. And so there's certainly things where a gene might cause an excess rate of speciation. No question about that. But whether your marker is that feature is another issue. Because if you simply give me an unbalanced phylogeny, it could be unbalanced simply by random sampling or by taxon sampling. You basically collected almost all your studies in South America. There's a bunch of equal species in North America you didn't collect. So you could have a biased phylogeny simply because how you sampled the taxa would give you an unbalanced phylogeny. Mutations which occurred in those early branches would give this signal of being correlated with the lopsided branch, but it's just simply sampling artifacts. So when you're declaring selection, you've got to be paranoid. What's that line? Just because you're paranoid doesn't mean they're not out to get you, right? And the same thing for declaring selection. Because everybody, you're working on a model organism, or your organism, non-model. You're invested in it. You want it. Your characters are interesting. right away you have an ascertainment bias. It's called confirmational bias, right? It's like certain people of different political persuasions watch certain news channels religiously to, to get the facts, i.e. to have people that think like they do say what they think, therefore locking them in. The same thing for your model organism or your organism you're studying. You're focusing on a trait, you're always thinking about how adapted that trait could be, and you're looking for those signals. That's okay, but just remember, you've got a bias. Anything you see you think might be a signal you're going to latch onto. So be careful when you're testing for selection to try to avoid that bias that we all intrinsically have. If you didn't have that bias, you wouldn't be a good biologist because you wouldn't care about your organism, right? So there's a trade-off. If you're a bad biologist, you don't care about the organism, you're probably less biased. But that latter is not an opinion of the Institute here, just my opinion. Okay, so what are the key points? Methods for detecting selection are prone to false positives, the rejection of the null, the equilibrium neutral model can occur for reasons other than positive or balancing selection, such as changes in population sizes. We'll talk about ways to look for that, and I'll use adjust in air quotes, but for most tests for selection, we can't put legitimate p-values on them. Rather, we simply extract genes which look interesting. Most are underpowered. Most selection events will be missed. If most adaptations occurs by polygenic sweeps, the vast majority of methods will miss those. The methods we have to detect those may not have enough power given the number of populations you have. We can detect only specific type of events. We can detect ongoing moderate to strong events. We can't detect weak events. We can't detect soft sweeps if we're using the wrong tests. We can look for repeated adaptive substitutions in a few codons over phylogeny. Those can certainly occur, but that's a unique class of events, right? That's not a representative random sample from how selection is occurring. Likewise, we can catch repeated adaptive substitutions over all sites in a gene. Now, again, I could have a gene with a fundamentally critical substitution. It's a substitution that made us different from chimpanzees. Single substitution in one gene. Its signal is going to be one extra substitution. You're not going to pick that up. So big events don't leave signals in divergence unless you've got repeatable events. So the bottom line is, this is all the reasons you want to be very cautious about making general statements about selection. If you detect selection, you're detecting a bias set. It's hard to extrapolate that and ask about selection in general. So, questions so far? That's my sobering slide. Yeah. Sure. So convergence, we can think about convergence at a couple levels. Number one is convergence at the level of phenotype. Lots of different ways to get there. That may not leave a pattern at the DNA level. We can also think about convergence in the sense that suppose you've got hemoglobin in several different species, and let's say in 2 species. And those 2 species are now put in, let's say, high oxygen tension environments. So they've got to fly over the Himalayas or something. Those may then have substitutions which occur, which are at similar sites. They may not be the same substitution. They may be at the same site, but I may get 2 different amino acids, for example. But you could get some patterns like that. But I want to be careful about convergence in the sense that you would pick those up in the right phylogeny, and you'd simply score them as multiple events. So in that example of looking over here, you notice when I've colored these, I haven't made those the same event. I'm just simply saying you've got independent mutations in these codons. They could be to the same site, that would be convergence, or they could be different sites. It may be, for example, that last box is a target for a pathogen. What you need to do is change it. You don't care what you change it to, you need to change it. That's not going to give you convergence as much. Or it could be you got to change that last target to a specific amino acid to avoid the pathogen. In that case, you'll see that last site showing convergent. But again, both those examples will appear as over the phylogeny, you'll have an excess of substitutions. And the phylogeny is critical because you basically have to know that those are two independent substitutions, not one mutation down here, then they gave rise to two daughter species. So phylogeny takes that into account to some extent. So you have to have a good phylogeny and a well-sampled phylogeny, right? You could introduce biases by how you sample taxa. We'll do a triangle here. Yeah, please.
I was wondering, so if you do a comparative study and you have, you can't assume that the species have the same effective population size.
Sure.
It might differ a lot. So, given that effective population size is so important for selection, like, can you say something about how that would bias your
Yeah, that's a great question. That's a really important question. So effective population size, I may have so a lot of things I was talking about in the lecture leading up to here are what happens when most of the selection coefficients are tiny and right about on the order of 1 over NE. In that case, small shifts in effective population size can take something that's neutral and make it deleterious, or vice versa. The things we're talking about here, we assume the selection coefficients are pretty big. So for NES is much bigger than 1 if, S is 10 to the minus 4 and your population size is 1 million or 1 billion. Those are both big and they're both completely determined by selection. So one way to think about it, when NE gets sufficiently big, the probably a new adaptive mutation is fixed as 2S. NE goes away. So effective population size is important, but it mainly comes in terms of asking, is the effective population size small enough? This adaptive mutation now isn't strong enough to be something other than effectively neutral. And for events like this, multiple substitutions, either over phylogeny or between species, we assume those events are pretty big. Yeah, and then we'll go ahead. So what are you finding frustrating? You finding my lecture frustrating, my style frustrating, or just aren't you? And you've exactly picked up on the important points. So basically, I'm the person that tells you that life's not fair. So people, when they see tests for selection, this kind of like the bright new shiny toy. This is wonderful and magical, and we all love it, and it's wonderful. And I'm kind of the harsh slap of reality, letting you know that these are important questions, and the methods have things to offer, but beware there are limitations on these methods. And so if you know the limitations going in, you can ask much more relevant questions, and you become much more of a cynic reading literature. You'll snicker a lot more in the library reading some of these papers. Like, yeah, sure. But so frustration is good, because it means you grasp the essence of the problem, which is this is not a magical solution. Selection is not easy to detect. We detect it by a sample, and even if it is there, you're not guaranteed to detect it. You're not in a happy place? Well, that's a good point. We can actually discuss that. We actually are in not that bad a place. So don't give up hope yet. Wait till I talk about, for example, estimating the fraction of adaptive substitutions. Right? So you'll get a little more, before I beat you down again, you'll get a little more upbeat. Then you'll get beaten down again. But your frustration is a good one because it means you're grasping some of the key points. Right? So don't be despondent about that. Just imagine it's good to hear about it now rather than putting in a massive NSF or NIH grant and get the reviews back. Other question? You have a question? No?
Me?
Yeah.
I did, but it's not particularly relevant.
That's okay.
You don't know what the ancestral state is in any of the other organisms in this phylogeny, so you don't really know if you don't have what was that site at that one place. You don't know that this is how it was structured.
The wonders of maximum likelihood. Not parsimony. Parsimony bad. That's because I'm a Felty student. So what you can do is you can basically, so if you have a model for character state evolution, and that model's not bad for nucleotides, we've got pretty good models for those. You can basically run over all probabilistic possible states and compute a likelihood for how likely those things are. So the idea is, it's a great question, the idea is you base your phylogeny not on the gene you're testing, right? Because lateral gene transfer occurs that can really mess you up. So you base your phylogeny on a large number of what you think are fair markers. You build that phylogeny, it's a very good point, build that phylogeny and then test your particular gene on it. Again, life isn't fair or easy. Other bad feelings I can, stoke up a little bit? Yeah. Yeah, so that's a great question. So one way to think about when you have something that's different from we've talked about, instead of just worrying it's different, ask what features would be different. So in a polyploid, what are some of the issues you'd face? Well, an obvious issue you face, but it's an issue we face, for example, in plants and diploids, is am I looking at an orthologous or paragolous comparison? That is, if I'm looking at, plants are full of duplicate genes. So if I'm looking at, let's say, a gene in Arabis and a gene in Arabidopsis, am I comparing two copies that trace back to a common ancestor recently, or am I comparing two copies that have duplicated and diverged quite a bit substantially? So I'm actually comparing, it's like comparing two beta hemoglobins versus comparing alpha versus beta hemoglobin. So there's always that issue when you've got duplicate genes. And that issue comes up to some extent in polyploids, because in a polyploid, you may have a situation where some copies are lost and some copies are not allowed to degrade, giving you effective diploids for some regions. So for example, tetrapods were tetraploid at one point. There's a gene duplication in the early tetrapod lineage. Most of those genes are now gone. There's little signals here or there. So we were an ancestral polyploid. Yes, we've lost most of those duplicate genes. But that means it can be tricky to reconstruct some of these phylogenies if you're not following a gene with a common evolutionary history. You're following genes whose evolutionary histories are a bit, reticulated. So that's one issue with polyploids. Other issues would be some of the summary statistics would be a little bit different, like levels of polymorphism, because you have to account for that. But those are things you can kind of adjust for. So the basic logic here would largely, the methods I've talked about, would largely extend to polyploids as well. Okay, so these are the main points. Again, this is the overview. Now let me kind of to walk you through quickly a couple of methods. We're going to go in those methods in much more detail. This is to kind of give you an idea about different ways you can use data to detect selection. So to detect ongoing selection, and basically ongoing selection is looking at polymorphism data. So I could, number one, look for excessive allele frequency change or divergence. Sorry, battery swap time. This green pointer is great, but it just sucks up batteries like you wouldn't believe. Number two is I could look for selective sweeps, a reduction in polymorphism. The thing is I need to have an outgroup to make sure that that reduction isn't due to reduction in simply the mutation rate. I could look for shifts in the allele frequency spectrum. For example, too many rare alleles or too many high frequency derived alleles. I could then look at allelic age inconsistency. An allele is too common relative to its age. How do you age an allele? We'll talk about that. Then we can talk about excessive LD in a common allele. A common allele under drift should be should be an old allele. An old allele means there's lots of time to react. So any LD block around that core sequence should be shrunk. A signal, what's called a long haplotype, an allele with lots of LD at a common frequency is a signal that is hard to generate by demography and therefore a pretty good signal for selection. So excessive allele frequency change. Logically, it's the most straightforward. The issue is we need estimates of effective population size in time to compute what the expected change is going to be relative to drift. I need 2 or more time points. I have weak power unless selection is strong or the time between sampling is long. So one classic example of this is to compare breeds from a common population selected for different goals. So here we have, here's your burger, here's your milkshake. So basically we've got Angus and Holstein. They came from a common ancestor. One has been selected for meat production, one has been selected for milk production, and several people have kind of scanned in here. The particular one thing I want to talk about, they looked at about 400 SNP markers on chromosome 19 and another 185 SNP markers on chromosome 29. What they simply did was they looked for allele frequency shifts where the shifts were higher in one direction relative to the other direction. You can adjust what amount of shift is significant by either doing a simulation study or by doing basically a randomization type of tests. And here's the result they got. Here is scanning across a chromosome. This is a sliding window of SNPs. This here is excessive divergence in the Angus direction, excessive divergence in the Holstein direction. Then the paper got fancy. See this little circle and all, wow, look at all those QTLs. The problem is this is very misleading. These are all kind of QTLs with respect to meat characteristics, QTLs with respect to milk characteristics. The problem is these confidence intervals are huge. A QTL confidence interval is simply 20 to 40 centimorgans. So, you know, basically these things fall in some huge uncertainty region, so I wouldn't get too excited about that. But this gives you kind of the basic idea. I take 2 populations that have been divergently selected. For example, I have an ancestral population that gave rise to, let's say, a highland population and a lowland population. I can look for excessive allele frequency shifts as I scan across those, and that's one indication of potential regions under selection. We'll talk a lot more about these. These are more formalized using FST type tests, that is Lewontin-Krakauer test. Another possibility is I could look for selective sweep. It's a classic visual tool. I can scan as usually presented. It's not a formal statistical test. It's just looking for a region of reduced variation. Positive selection tends to reduce NE, reduces time to recent common ancestor and variation. Balancing selection increases effective population size and therefore increases variation. So I give you examples here again. With balancing selection, I get a longer time to common ancestry, a chance for more mutations. With selective sweeps, I get a shorter time to common ancestry, time for less mutations. We typically do these by using a sliding window. If I look at a single polymorphic site, it's really a lot of noise, very noisy. So I just take a collection of them and slide that window across and basically plot that. And so what I basically would expect is that it should, we can visually look for that. There's a class of likelihood-based tests called Sweep Finder and the composite likelihood ratio test, which basically ask, is the form of that reduction consistent with what I expect? And the way you get from that is I give you this expression for F, which is reduction, which is a complex function based upon the recombination and selective strength. You can actually build that in likelihood, and that basically allows you to estimate the selection strength and also where the sweep probably was. So again, you see in these pictures, a decrease over background is consistent with directional selection, an increase over background with stabilizing selection. Here is maize and teosinte. Here's modern maize. Here's teosinte with this multiple branched. There's a mutation that takes maize and turns it into the teosinte architecture in terms of multiple branched. It's called teosinte branched 1. It's an obvious candidate gene for domestication. As I mentioned, when people scanned that candidate gene, what they saw, the gray down here is maize, the solid is teosinte. Maize is typically under teosinte because population bottleneck to form it, but over here it's quite dramatic. So this reduction over here could simply be low rates of variation, low rates of mutation, but you see very high levels in teosinte, which kind of rules out a reduction in mutation rates in this window unless something magical happened to reduce mutation rates in there in maize versus teosinte, which is kind of unlikely. I gave you the ADH story again where you see an excessive level of variation. Here's another one. So this is Drosophila. Remember, Drosophila melanogaster is a commensal species. It followed humans out of Africa. And so one thing you can then do is look at the nucleotide diversity in Africa down here at a given locus, look at the nucleotide diversity in Europe. What you basically see is, for example, this locus over here had a nucleotide diversity of about basically 20 SNPs per kilobase. You go over here, nucleotide diversity is about 4 SNPs per kilobase. What you see is all these points are less than, more diversity in Africa than in Europe with some minor outliers. You expect that because it's a bottleneck population. What you don't expect are these things down here which have no variation in Europe, but which have rather large variation in Africa. So these solid points down here are targets for genes which have seen a sweep. When you move out of the environment in Africa into the new environment in Europe, you have an environmental swap, you could see effects. Obviously some things down here could be as well, but the ones with zero variation, given how much they have here, those are pretty good consistent samples. So again, that's kind of a visual test. We'll talk about more formal statistical tests a little bit later on, but basically looking at levels of divergence and trying to correct for the fact that it could be low polymorphism. Using two populations here, this corrects, showing you have high levels in Africa, but low levels in Europe. Here's another example. This now is on soft sweeps. This is double muscle cattle, the Belgian blue. It's an enormous thing, and it's a myostatin mutant that basically gives it extra muscle. In many countries, these are outlawed because they're so big you have to give birth by cesarean section. So myostatin mutations basically give you excessive muscle development. They're called double muscle, and there are basically 3 breeds of cattle that do that: Blue, Valais, and Piedmont. And so what this group did here was they looked at microsatellite variation. So let me remind you what microsatellites are. You have regions of DNA that have multiple repeat short copies, like little train cars. When you've got multiple repeat short copies, they can mutate at high rates, not from mutation, but when you have multiple short repeat copies, when DNA is replicating, the polymerase slips. So the error rates in terms of copy number for microsatellite are on the order of 1 in 100 to 1 in 1,000. The mutation rates on a SNP are on the order of 1 in a billion. So microsatellites have far higher mutation rates, and therefore they show a lot more variation. If you ever watched a crime scene show, the 13 CODIS markers they use to give a 1 in 20 trillion chance of matching someone, it's based on 13 markers, but they're microsatellites. They each have on average about 10 loci, each with about frequency of 1/10. So microsatellites are highly polymorphic markers. If you see reduction in variation in microsatellites, it's a good indication things have come from a relatively recent common ancestor because they show so much variation. So in the old days, before SNPs were easily available, people would often look at microsatellites because reduction in microsatellite variation relative to some standard indicated a reduction in the affected population size. So what people basically did was they looked at a number of microsatellites, and they noticed that as you got near the myostatin gene, the microsatellite variation decreased. It didn't go to 0, and it was kind of asymmetric. You expect the variation to be, kind of symmetrically distributed, reduced around the myostatin. It wasn't. And again, as I mentioned, sweeps are often asymmetric about the point where the actual sweep occurred. What they also noticed, though, was that in Belgian blue and Piedmonts, there were not as much reduction. And whereas in this breed here, there was a tremendous reduction. And the authors noted that until the 1950s, the 2 other breeds here were dual purpose, both milk and meat. And there were records for the phenotypes that matched the double muscling that predate World War I, and therefore predate the intensive selection on here. So the fact there's not a strong signal around myostatin in these 2 breeds is probably soft selection. The allele was existing, And the selection wasn't just for meat, it was for meat and milk. Conversely, in this breed up here, that's a more recent breed, and the mutation, there was no evidence of this mutation until they started selecting for meat alone. So you have a fairly strong signal in one breed around an obvious, a known target gene, and weak signals in the other two. Again, this is probably soft sweep, and this is the hard sweep. The mutation was pre-existing in these two breeds when they were being selected for multiple purposes. The mutation arose in this breed after it was selected for a specific purpose. This is a nice agricultural example of where you get a hard sweep or a soft sweep signal. So what are the issues with using sweeps? You need sufficient background variation before selection to detect a sweep signal. So if your background level is here and your domestication brings you down here, you need to be enough room so you can catch a reduction in polymorphism. That's easy in maize. In sorghum, your background levels like this is much more difficult. So in things like the cabbage white butterfly, which probably went through a bottleneck, that background level coming in may actually be rather low, and therefore depressions in that background level would be even harder to detect. So the issue here is you need sufficient background variation for a strong signal. Likewise, inbreeding reduces that variation as well. When you have a signal, it persists for a short amount of time. Oftentimes, the type of signals persist for only about 1/10 NE of a generation, and the effects basically scale like this. So if selection is weak or recombination strong, it covers a small window. As I mentioned, the sweep regions are often asymmetric about the target site. You simply can't take a sweep region and take the middle of it and say that's where the target site is. Hard sweeps can be detected, whereas soft sweeps leave at best a weak signal. Many tests can detect hard sweeps, will miss soft sweeps entirely. Tests for soft sweeps can miss hard sweeps entirely. So you have to apply the right sort of test. You also can't go what I call test shopping. You have a region of interest, you apply 40 different tests until you get one that has a significant result. You've got to do multiple comparisons to adjust for the fact that you're doing multiple testing. Again, I mentioned hard sweep versus soft sweep, gives you the idea. So again, we'll go in detail about those, but the idea is we use the pattern from a sweep of either suppressed variation or the specific structure, the curve of that suppressed variation to basically look. That specific curve gives rise to a class of likelihood tests which you can actually put p-values on. A second approach is site frequency spectrum tests. The site frequency spectrum simply says, what's the fraction of sites that have a singleton, 2 copies, 3 copies, 4 copies. You can either do an unfolded, so it's 1 derived copy, 2 derived copies, or do a folded, the minor allele frequency has 1 copy, the minor allele frequency has 2 copies. And the idea is the site frequency spectrum is entirely a function of theta under drift and neutral and equilibrium. So you can use different aspects of that site frequency spectrum to estimate theta. You can use the number of singletons, you can use the diversity, you can use the number of segregating sites. Those are all attempts to basically look at single measures. You can then contrast those measures and ask if they're consistent. That's what a lot of the site frequency spectrum test does, such as Tajima's D. The problem, a significant result can result from selection or from the fact you've got population structure that is drifted mutation isn't in equilibrium, or you've got a structured population, so you have migration. So a population bottleneck will give a similar signal into Tajima's D as a selective sweep. A structured population will give a similar signal into Tajima's D as overdominant selection. Yeah, question?
Could you talk about those demographic models that people use to try to include?
We will. We'll talk about those in detail. Yeah. And the answer is, if you have the right model tested, they work, but people often don't test allelic surfing. They assume a static, they don't assume a static size, they assume a static space population. Allelic surfing, the edges of that population have different behavior. If you don't put those in, you can have problems in your simulations. So basically, here's the rough idea. So again, if theta equals 4 NEmu, that's our measure either over a site or over a region of the variation. If I look at the number of segregating sites, I can just count them up. Segregating, 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 done. Don't care what the polymorphism is, just segregating, non-segregating. The expected value of that equals this coefficient here, which is simply a function of the sample size times theta. The average pairwise difference, its expected value is theta. The number of singletons. So singletons here, I'm using eta there. That means I'm using the unfolded spectrum. Sorry, I'm using the folded spectrum. So basically, the number of copies that appear, sites that appear as one copy, is theta times, again, this thing that depends upon sample size. I've got 3 different estimates of theta. Here are their sampling errors. They should all be consistent if the neutral model holds. So Tajima's D compares pairwise differences in segregating sites. There are other tests we'll talk about that do other comparisons and also other measures of the site frequency spectrum. So here's Tajima's D. It's like a t-statistic, mean 1 minus mean 2 over sampling error. Sampling error basically follows from here and from the covariance structure between these two tests. So there's the formula for it. A negative value means I have an excessive number of rare alleles. Why? Well, if you look at pairwise differences kind of look at common alleles. Segregating sites look at rare alleles. So if I've got more segregating sites, then this is going to be larger than the pairwise differences. A positive value means I've got an excessive number of common alleles. That's consistent with either balancing selection or population structure. So a Tajima's D value by itself for a single locus doesn't tell you much. What people typically do is they have a whole bunch of test candidates. They compute Tajima's D on all of those. Then they construct a histogram. Then they say, look, this value is an outlier. Well, you know, that's not a p-value. It's an outlier. So we'll talk a lot. We have basically almost a half a lecture on tests based upon site frequency spectrum, but that's the basic idea. I have different measures that summarize aspects of the site frequency spectrum. I compare estimates. Those are basically looking at different parts, and I can see if they're consistent. If I've got the equilibrium neutral model, they all should be consistent. If I see departures from that, that model is not correct. It's usually not correct because the equilibrium assumption has failed. It may also, on top of that, also fail because the neutral assumption has failed. Another category, again, this is an overview. We're going to go back and talk about these things all in detail. Another category of tests I talked about very briefly was allelic age tests. And basically, here's the expression that says, if I take my allele frequency x, what's the expected time? There's a huge sampling error on here, but the bottom line is that underdrift, a common allele is an old allele. So common alleles should not be young alleles. So if we can age an allele, and there's a couple ways to do that. Number one, the allele is defined by a certain SNP. That SNP may arise on a certain background initially, and I can look for how long that association persists. If I know the recombination fraction, I can look at the association that's there, the recombination fraction, and basically age the allele that way. I can also age the allele by looking at the blocks of LD, right? Or I can age the allele by looking at if it's right next to an STR. The older the STR is, the more variant, the older the allele is, the more variance I should see at the STR. Remember, that allele arose on one chromosome and had one particular allele at the STR around it. As that allele ages, that STR then acquires mutations and has a copy number variance or a spread variance about it. So we can use additional information from linked site to age an allele independent of its frequency. So allelic age tests basically ask, are the ages based upon frequencies and the ages based upon some other measure consistent? Now the key is these things have huge sampling errors, so they've got to be really inconsistent for this idea to work. And here's something we did through before. Remember the del32 deletion in the CCR5 receptor that gives you significant resistance to HIV. We've shown based upon a frequency of 10%, effective population size of 5,000, that this allele should be, if it's neutral, about 5,000 generations old with a pretty large standard error, from about 600 generations up to about 15,000 generations. Standard errors on these are really big, but nonetheless. So the other way we can age the allele is this mutation is in strong disequilibrium with allele 215 at an STR marker. So that 85% of all the sample of the 46 sampled delta mutations had this allele. How would that happen? Basically, when that allele arose, here it is, delta, on that same chromosome, over here, I have the 215 allele at the AFMB site. So initially, that was the only chromosome type that was present. It then gets randomized by recombination, but when we go ahead and sample, 85% of the population still has this haplotype. This is basically decay of linkage disequilibrium. So we can then use the recombination frequency between them, the fact this is at 85%, to actually ask how many generations would it take if you'd only have this association be 85%. You can go ahead and do that, and it's 28 generations. 28 is a lot smaller than 5,000. Even if you take the lower value of about 600, it's still far, far stronger than that. So people took this huge discrepancy in allelic age based upon frequency and allelic age based upon an independent measure of age. They took that great disparity as an indication there's probably strong selection on this site. You can actually then use it to take 28 generations and this allele frequency, ask how long it would take selection to drive you up there. And the selection coefficient was pretty high, I think around 5%. Yeah? So if that happened, then delta would be on some other background. And so you would see delta on that other background as well, right? So for example, so here is a chromosome with, let's say, 214, and here's a chromosome, let's say, with 213. If delta arose on here, most of the copies of delta then would be associated with 213. The fact that most are associated with 215 means it arose on this background. And then you have, and you can build, you can make models that basically adjust for that as well. This is kind of the first approximation. But the key idea here is you can age an allele by looking at how quickly recombination decays with respect to a known marker. Because when that allele arose, it arose as a single copy. If it arose as multiple copies, that's a separate issue. If it arose as a single copy on one background, you can then figure out the background and figure out the decay rate, and then by the decay rate, you can figure out the age. So maybe it's not 30, maybe it's 10, maybe it's 50. That's still quite different from the way lower range on that of 600. So the idea is we can take DNA sequence information and use that to age an allele. We can then, because you take the alleles defined by a certain SNP, you then look at that, a SNP, either a linked STR, how variable is it? The more variable the STR, the older the allele. You then look at linkage disequilibrium. There are several ways you can age the allele from sequence independent of its frequency. Then you can compute what you expect its age to be based upon the frequency. This is the one fraught with the most error, and then look for inconsistencies between these. So it's not a very powerful approach, but it's a pretty logical approach. Long haplotype methods use this idea, but put it basically on steroids and make it on a statistically much more rigorous framework. So long haplotypes is this notion that as an allele becomes older, again, an allele initially appears on a background, and then there's a big linkage block, a big block of linkage disequilibrium around that. As that allele becomes older, that block decays away by recombination. Remember that the disequilibrium at time t equals disequilibrium times 0 times 1 minus c to the t, which is basically d0 e to the minus ct. So as time gets larger, that block of disequilibrium gets smaller. So what you might expect is if this is time, an old underdrift, an old allele, a common allele is an old allele. An old allele has a small block of LD. So a broad class of very powerful tests basically ask, do I have common alleles with large haplotypes? You basically contrast the 2 or more alleles at a site to control for local recombination rate, but the basic idea is common alleles with long haplotypes are a good signal for selection, and they're rather robust against demography. Not completely, allelic surfing can generate this. On that wave, if a new mutation arises on that wave can then sweep that allele up to fairly high frequency fairly quickly and give you a signal like this as well. So even though this is a good signal, it's robust to many demography issues. It's not robust to allelic surfing. Allelic surfing is always the issue if you have an expanding population. I can always say the signal you detected was a rare event that occurred on the wave of this, and that generates a coalescence structure very similar to directional selection. Yeah. If you have haplotypes, you got to phase it. Now, the thing to be careful of is nobody talks about this, but typically people use programs to phase their data as compared to phasing it directly by sequencing technologies. Those programs make assumptions. No one, as far as I can tell, has ever asked what the effects of selection are on those phasing programs. So if you phase the data using those programs and then run a selection analysis, I'm not sure how good that is. I'm not saying it's bad, I'm just saying that's something I'd be concerned about, and as far as I know, no one's checked. I actually asked Matt Stevens, who wrote the big phase program. He said as far as he knew, no one's looked at it, because it's not that much of an issue. But phasing with technology now is an easier issue than it was a while ago. It simply depends upon your read length and stuff. Again, these are things we're going to go back and talk about. I'm trying to give you the big picture, so when we go in all the detailed tests, you can still kind of look back and get that sort of mile-high view. So those are examples of a few of the tests we'll talk about using polymorphism data. Polymorphism data says I take a sample from a population, or I take samples from several populations with a common ancestor, or one population at several points in time, and I look for features that suggest a pattern that's different from what I expect from drift. Those are detecting an ongoing single event. I'm now moving to the class of tests that take divergence data. You have a model species, Paris rapi. Take the cabbage white, take Paris pertataceae, the checkered white out here, Paris occidentalis, sibling species, and sequence rapi, sequence occidentalis, get an estimate of polymorphism in one of those two, and then ask if there's some inconsistency in the pattern. And this approach is Hudson-Crichton-Aguade HKA or McDonald-Crichton MK test. The nice thing is McDonald-Crichton, as we'll talk about, in some respects is rather robust to demography. In other respects, it's not. We'll talk about that in detail. This requires polymorphism data for one or both species and divergence data between species. And here's the logic. I look at a given site, let's say synonymous sites versus replacement sites. Those are going to have different effectively neutral mutation rates. The idea is you have a mutation rate and you basically have two types of mutations. Advantageous are so rare we're going to ignore them. You've got deleterious mutations. Those are assumed to just get wiped out right away and never contribute. Then what's left over is neutral. So obviously, if the actual mutation rate at silent and replacement sites is the same, the effective mutation rate is much higher at silent sites than at replacement sites because a much higher fraction of mutations at a replacement site are deleterious. So you'd have, we'll call I the mutation rate at a site. The expecting level of heterozygosity is just 4Ne mu I. The expected divergence is just 2T mu I. So the ratio of heterozygosity to divergence, hey, look at that. The mutation rate differences cancel. So if I look at the ratio of heterozygosity to divergence at silent sites, heterozygosity to divergent at replacement sites, those ratios should be the same. It's a simple chi-square test, or Fisher's exact test, which is the chi-square test when your sample values have an expected value less than 5. Fisher is, of course, R.A. Fisher. And that's a slightly better way to do that. R does the Fisher exact test for you. So here is the original paper. It's a very cool paper. 1991. Yeah, so I guess I mentioned, so Kreutzmann was 1999 on 2000. He examined ADH, alcohol dehydrogenase, in melanogaster and in simulans. What he basically had was he had a small sample, I think it was 11 alleles for melanogaster, and he saw, he compared melanogaster and simulans. He saw 24 fixed differences, 7 were replacement, 17 were synonymous. He saw 44 polymorphic sites segregating, 2 of which were replacement, 42 were synonymous. So here's the data. So if you look in, I think it was melanogaster, he saw 44 polymorphisms, 42 were in synonymous sites, 2 were in replacement sites. He looked at the difference between melanogaster and simulans and saw there were 24 fixed differences, 17 in synonymous, 7 in replacement. Well, if you look at that, that's a 20-to-1 ratio. That's a little bit more than 2-to-1. The Fisher's exact test gives a highly, highly significant P. There's a significant lack of fit. So there's an excess number of substitutions relative level of polymorphism. Now that could be because the polymorphism is too low or the replacements are too high. It's a relative concept. But the key thing here is you can estimate the number of adaptive substitutions. How cool is that? So what's the, if I just use the polymorphism data here, that's the polymorphism data, and the synonymous level over here, I can estimate the number of replacement sites I should see based upon this polymorphism ratio. So basically take this and multiply by that. So I basically would expect to see on the order of, that's roughly 1 replacement. I see 7. That tells me I see 6 excessive replacements. So what's the rate of replacement substitutions there? It's 6 divided by 7. It's pretty high. So this approach we'll talk about, I think, on Wednesday, about estimating the fraction of adaptive substitutions. We'll talk about problems with that approach. We'll also talk about what the data seem to show in that. So this is, it's a super simple idea. It's robust in that if I have a non-equilibrium population, it doesn't mess up that polymorphism ratio. Why? Because basically I've got a value in both sides that cancels when I take that ratio. So it's robust to issues in dermography, but the problem where you can arise is, suppose the effective population size during the polymorphic phase was different from the effective population size during the fixed phase. If that case happens, the effectively neutral mutation rates can differ. And as we'll see, that can compromise the test. Yes? Mm-hmm. Yeah, and a big issue we'll talk about at length is if you have deleterious alleles segregating, they will contribute to the polymorphic count, but they'll never get fixed. So that will compromise the test. We'll talk about ways to get around that. Sure. That would do it. The key is basically what you're trying to do is the polymorphic sample should be representative of the process that gave the divergence as well. Yeah, and the nice thing about Macdonald-Kreutzmann is it's pretty robust and simple, but as in with all tests, it's not without problems. But it's probably it's probably the test that is probably most problem-free. It's not perfect, as we'll see, by any means, but it's a very simple test that basically does the job generally fairly well. And HK is basically a modification of that. HK actually came out before that. Other questions or comments so far? Again, all the things I'm talking about we'll cover in more detail a bit later on. Just trying to give you a quick overview to give you some basic idea and make this notion of detecting selection, which sounded really abstract when we talked about frequency spectrum and sweep theory, trying to make it a little bit more concrete before we get down to the details. So strengths and weaknesses of Macdonald-Kreutzmann and other such tests where I detect divergence, it only detects a pattern of adaptive substitutions. It requires multiple events to have any power, and I can't tell which replacements were selectively driven. Here, for example, I know that 6 out of the 7 were, but I'm not sure which one. I'm not sure which one would be the neutral one. And again, this is an average. It may be there's 3 neutral and only 4 adaptive. It's robust to demographic issues, but it's not foolproof. I've got to mention this here. So any change in the constraints between the process generating polymorphism and the process generating conversion can be regarded as evidence for selection when it's not the case. Let me give you a concrete example here. So this is the melanocortin receptor for skin pigmentation in humans. So here is a polymorphic sample of humans from Africa, 4 silent, no replacement. Here are fixed differences between human and chimp. If you go ahead and run that, what you see is it's not quite significant, but awfully close. And it looks like there's an excessive number of replacement substitutions between humans and chimpanzees given your polymorphic sample in Africa. Pretty convincing, right? Looks good. However, let's take that same data, human chimpanzee, and now have my polymorphic sample be individuals from Europe and East Asia. I see 13 polymorphisms. Now I have a very non-significant p-value. Why? I've got no replacement in Africa, polymorphic, significant replacement over here in Europe and Asia. Why? Relaxation of selection constraints. Individuals evolving in Africa don't have the melanocortin gene. They have huge amounts of UV exposure. By the way, the same thing if you take Asians from Papua New Guinea and India. You get a pattern like this, a very little replacement polymorphism, whereas elsewise you get a lot. So the idea is I have a removal of selective constraints. Here, you can't have any replacements because you don't make a functioning melanocortin, and that's very problematic. Over here, you have less damage of high sun level, and therefore there's a relaxation of selective constraints, and this signal goes away. The question is, what's the right answer? Divergence data hasn't changed. Your reference polymorphic sample determines what's correct, and there's no right or wrong answer here. This simply shows that where you take your reference is very critical. What this clearly shows is strong purifying selection in Africa, weak purifying selection in Europe. Was there strong or weak purifying selection in the human-chimp split? We don't know. So you can't make a definitive statement. This is a good example to point out one issue with McDonald-Kreitman is your polymorphic sample really determines your result. And here, 2 logical choices of polymorphic sample give me very different conclusions. Neutral changes, strong selection. Yeah. You could do that. That's one way to check for your robustness in that. But the point is that you could even have this. So for example, suppose that you had weak selective constraints in your polymorphic sample, but strong selective constraints during your divergence period. You'd never pick that up from one sample or multiple sampling. So this just points to what you can happen from within a sample. You can also have this same thing about selective constraints being different within a population versus between populations. And so you could get some robustness by looking at different samples, and that would tell you if you had a problem on that time scale. But if that result didn't give you a problem, you may still have a problem overall, because you can't sample that historical time when the divergence actually occurred. So McDonald-Kreitman is nice and powerful, but there are issues involved. Yeah?
Does this also mean that your choice of reference population affects the question you're asking?
To some extent, yeah. I mean, it affects the test, so by definition, it affects the question you're asking. Right? The idea is your reference population should be representative of the constraints you expect doing your divergence. And you know, which of these is better, I have no one has any idea. You could make it, determining which of these is better is just making up your answer and then justifying it. The point is McDonald-Kreitman has this issue. We'll see an example later on where I'm looking at 2 sunflowers. If I use sunflower 1 for my polymorphic sample, I see strong evidence of adaptive substitutions. If I use sunflower 2 for my polymorphic sample, the same divergence data, I see no evidence whatsoever. What's the correct result? We don't know. Just be aware that you have this issue with McDonald-Kreitman tests. The last category of tests are Ka to Ks tests. And people use several different things. So Ka for amino acid is the substitution rate at replacements. So I look over phylogeny and I ask, at what frequency do I see changes that result in changes in amino acids? Then I look over that phylogeny and see at what rate do I see changes in the DNA sequence that give me synonymous, that is, are silent. So Ka to Ks ratio is often called omega, or the acceptance ratio. A Ka to Ks ratio bigger than 1 is often taken as bulletproof evidence for positive selection. Close, but not quite. The problem is, most codons have Ks bigger than Ka, so even if I have adaptive events occurring within a gene, certain sites that signal get swamped might consider the entire gene. Let's look at the classic example of that. Do I have? No. The classic example is Masatoshi Nei and Austin Hughes. They looked at the major histocompatibility class 1 and class 2 loci. If you compare mouse and humans, you see an omega ratio that's significantly less than 1. Ka to Ks is much less than 1. However, they had magic. And the magic was the three-dimensional protein folding sequences, so they knew which residues were involved in antigen binding. And the idea is those residues should evolve fastest. Now remember, think of a protein as a rope. What matters on the rope is not distances, but how that rope folds. So sites very distant from each other may be in very close contact. So you can't simply look at physical position on that linear sequence and really say much about what points are in contact with which other points. So you can't just simply, take chunks on there at random and ask about them. The advantage they had was they knew how it folded, and they simply, get your question in a second, they simply took those folding sites and looked at the Ka to Ks ratio of those folding sites that made in contact, that made antigen binding, looked at Ka to Ks over everything else. At the folding antigen contact sites, Ka was much bigger than Ka to Ks was much bigger than 1. At all the other sites, it was less than 1, significantly less than 1. And the advantage they had was they knew a priori which sites to compare from the protein folding structure and some idea about what they do.
So a couple times you talked about local versus distant.
Right.
Is that linear distance or folding?
Yeah, good question. So local versus distant, what I was talking about when people look at gene expression data and try to map QTLs involved in gene expression, historically, if I had a target gene and my QTL or GWAS hit was right next to it, they would call that a cis effect. If it was further away or on a different chromosome, they'd call it a trans effect. That's bad terminology because cis and trans mean specific functional things, not specific distant things. A cis means that I've got a binding site that affects everything on that chromosome. A trans thing means I make a trans factor. So the terminology now is to go with local, meaning that eQTL hit very close to the gene, and distant. So I'm talking about distance and not confusing the cis-trans functionality with distance. Same thing, I mean, the idea here is somewhat similar on proteins in the sense that I simply can't look at a linear sequence and assume a block of things all interact, because they may not. Proteins fold in really weird, funky ways. You need the 3D structure and some idea about what the protein does to know which residues to pick out, which residues to ignore. So basically what they're doing here is this is a codon by codon test. They took the codons and they lumped them into two categories. Those involved in antigen binding sites, those that aren't. We can take this idea and apply it over to a phylogeny, if it's big enough, and look at codon one at a time. Here, they simply had two species, but they could group codons into two different categories based upon functional reasoning, and then they'd use that functional reasoning then to test those two different categories. So this is multiple substitutions in the same codon over time. The extension of this are these codon-based models. So if you look at the entire gene, even in MHC, that ratio is less than 1, the Ka to Ks ratio. even though specific residues were much bigger than 1. Well, we knew those residues because we knew something about the biology, the three-dimensional folding and the binding sites. You don't know that for a random protein. So how do you get around that? One way are these codon models. So basically, you assign all non-stop codons. There's 64 codons, 3 are stop. So you basically assign them 1 to 61, rank them. Then you have a matrix that says transmitting from one codon to another, you assume a single mutation. And what you basically allow then do is a model where the transition probabilities between all 1-nucleotide transitions are constructed. You then put in a parameter that asks, does the model assume omega 1 or omega bigger than 1? You get something that looks like this in the simplest thing. It looks complicated, but here's the idea. So here is your codon. It can jump to 1, 2, 3, 4, 5, 6, 7, 8, 9 alternative steps, maybe 8 or 7 if one's in the stop codon. You allow transitions and transversions to have different rates, so you account for that. These terms here account for codon bias, and the omega terms here basically account for, that's the Ka to Ks ratio. Everything with a star gives you a different amino acid coding sequence, so that would be a Ka, 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 and only this one over here is a Ks. So these things have the omega on them for that extra ratio. So basically then have a huge model with all these codons, fit the model with maximum likelihood, and then basically test is there evidence that omega is bigger or less than 1, and do it on a codon by codon basis. So it's elaborate and involved, but there's a lot of work in here, and it seems to work so-so. So you can make, here are the transitions, you can make them, you can put omega into 3 categories. So you could have a, what's called a mixture distribution. With some probability, omega is 0. With some probability, omega is 1. With some probability, omega is bigger than 1. You can then estimate those mixture probabilities. You can make your model as complicated as you want and estimate things. The problem is the likelihood services may be very broad. So your mixture may say that the weight on the selected class is 5%, but that 5% could range from 1% to 18%. It can be very varied. You can also use Bayes' theorem, I'll change that approximation, to basically then ask if you get a positive, if you get results that some fraction of omega is bigger than 0, you can then basically ask, is the specific codon under selection, and then return a posterior probability for that. So that's the third category of these codon models. And an example here is Erebus. I think I mentioned chitinase genes. When they go ahead and fit those chitinase genes, this is Tom Mitchell-Ols's lab, they estimated that between 65 and about 80% of the replacement were deleterious, and between 5 and 14 were advantageous. Among those advantageous, the Ka to Ks ratio is estimated to be about 7. They then basically looked at where those sites may be. Here's the protein, and basically the sites in color are the sites that have high prior probability, and a lot of them fall in this clef here, which is thought to be the major action. So again, the details on that aren't important. The idea is you make this elaborate model, and right away you should be a little suspect. Elaborate model means you're fitting a lot of things. Do you have enough data to fit a lot of things? That's always an issue with these likelihood models. So what are the strengths and weaknesses of these codon-based models? You can assign repeated selection to specific codons, and it requires only single selections from each species. The weaknesses are models can be rather delicate, to put it mildly, and you can only detect repeated selection at particular codons, not throughout a gene. So if you have a pattern of excess substitutions throughout a gene, it might not give you enough signal when you go through on a codon by codon basis, because it's not amalgamating changes overall the individual codons, it's doing it on a codon by codon basis. And that leads us to this discussion here. Very famous paper by Gould and Lewontin called the spandrels of San Marco. And the idea was, man, look at all that detail. It must really do something important. So you have a beetle, and that's the structure on the sheath wing of your beetle. Look at all those elaborate ribs. Boy, evolution must really work hard to get that. That must be something really, really important. And the punchline from Gould and Lewontin was elaborate structure does not imply function or adaptation. A great modern example, actually it predates this paper here, there's a very classic paper that people should read about genomics. It's an old paper on Boolean gene networks in Science by Stuart Kaufman. And what Kaufman did was something really simple. Yet this is, really old. He probably even used like Fortran to program this. He basically had a simulation where he had a bunch of light bulbs, genes, in his computer. He randomly threw down connections between them and randomly put down rules. Like the rules are, if one is on, then one is on. If one is on, then two is on. If one is on, then two is off. Simple binary rules. He threw down this random network, threw random rules down, and then randomly set the state and let it run. What happened was whole sections lit up and went off simultaneously. So if you saw that, you'd say, look at all that evolved coordinated gene action. It's all random. And you don't see it unless you do the simulations to see how elaborate these things can be. So you really need null models when evaluating evolution. So the key thing here is that elaborate structure does not imply adaptation, does not imply function. I think I showed you the first day that beautiful Rothschilda cinctus silk moth with the clear windows, red moth with the clear windows in its forewing. Those clear windows could be there simply because by random scales weren't put down there and it had no fitness consequence, so no big deal. So elaborate structure does not imply function or adaptation. This is more for people with a molecular bent. So if you're a molecular biologist, I often give this talk in like to stir up molecular biologists, this slide. You just spent your whole, a whole grant cycle or more looking at what a particular molecule does. Since you're looking at what it does, it clearly in your mind has a function, and therefore, since it has a function, it has a strong history of adaptation that shaped its function. You cannot assume an adaptive explanation just because the structure is complex. And a great example of that was a paper that if it were on any other trait would have been in the textbooks as a classic case of showing selection. But the trait was human intelligence. And one way to look at this was the alleles that caused this were at lower frequency in Africans and higher frequency in Asians and Europeans. And obviously that got people very upset, so they drilled down more carefully. If this would have been about butterfly wing color, they wouldn't have done this, it would have been reciting. So let's go through and look at this. There are genes in humans, microcephalin and ASPN, abnormal spindle microcephaly associated, where basically if you have mutations in either one of these, instead of having your 1400 cubic centimeter cranium, you have a 400 cubic centimeter cranium. And that's comparable to the size of early hominoids. So not surprisingly, people looked at those genes in the primate lineage. And what they saw were a Ka to Ks ratio of 1.3 to, an excess ratio leading, is that microcephalin? So on both microcephalin and ASP, leading to higher rates. These are basically the omega rates, Ka to Ks. Higher rates in the great apes. Bigger than 1, bigger than 1, significantly bigger than 1. Suggesting that ASPM have under selection. A similar thing was found for microcephalin. So evidence in the phylogeny that for whatever reason, genes involved with larger cranial size were selected for. That is, they had excess number of replacement substitutions than you'd expect by chance. Okay. You then went and looked at a McDonald-Kreitman test on examples in human populations. Here's what you saw, polymorphic sample, and here's human chimp. That's highly significant, p-value less than 1, and the excess is 15 replacement substitutions. Human and chimp, 15 extra substitutions in a gene involving cranial size. Of course it'll be in Science or Nature. So then people took that same data. This is for an adaptive human chimpanzee divergence. They took that exact same data and then they looked at segregating variants in here. What they found was extended linkage to disequilibrium and small within allele variation. So they basically estimated they had long haplotypes, estimated they're under strong selection. Extensive coalescence simulations under a variety of population structures. And this work was done by Dick Hudson, the simulations, who was the leading guy on these simulations. They couldn't generate this pattern by drift. That was in microcephalin, ASPN, exactly the same pattern, a common haplotype with long LD and a very recent estimated origin relative to its frequency. And again, drift didn't regard this. So because this study showing evidence of selection in the great apes, evidence of excessive replacement in going from humans to chimpanzees, and evidence of apparent selection in the human population, these long haplotypes, what got people excited was the putative adaptive haplotypes were higher in frequency in Asia and Europe than Africa. So people got really excited about that and went in and actually did the studies where they basically took individuals with these different length haplotypes and gave them some generic measurement of intelligence. Not very good, but the bottom line, absolutely no difference. So if it weren't for the fact that people were worried about people making inappropriate social statements from this data, it would never have been looked at as carefully. And we regard it as a classic example of selection in the great apes, human chimpanzee, and ongoing in the human population. Why these unusual things? Well, two explanations. Number one, by the way, really large sample sizes to check these. Number one, it's under selection for some reason other than intelligence. Very possible. Number two, allelic surfing. Europe and Asia are a migration coming out of Africa. If you had mutations in that, they'd be edge of that migration. As that migration expanded forward, they'd have a coalescent structure extremely similar to a gene under selection. So this is the big cautionary tale. If you look at all the pieces, it looks fascinating and interesting and integrated, and yet when you put it all together, it falls apart. So if you were frustrated earlier, good thing you're not an anthropologist working on cranial size, you'd be very frustrated there. So that's kind of the overview of tested selection using sequence data. We're now going to drill down and look at those various categories in more detail. Polymorphism, allelic divergence, site frequency, allele frequency, haplotype test, McDonald-Kreitman, HKA, codon models. That's basically over the next 5 or so lectures. So questions before we proceed? Mm-hmm. Yeah, so you bring up a great point. One of the problems that makes McDonald-Kreitman difficult to apply, if you have slightly deleterious alleles, they will inflate the number of polymorphisms you see, but they will never be substituted. And that decreases your ability to detect selection. So one of the big issues is how do you adjust for that? We'll talk about that. People have this rule about arbitrarily throwing out singletons. Why? And we'll talk about some methods people have suggested to get around that idea that if you've got deleterious alleles segregating that are mildly deleterious, they'll contribute to polymorphisms at low frequencies, but they'll never contribute to substitutions, and that depresses the number of excess substitutions you see. Yeah? It does, but the bottom line is you can think of it kind of as sample size. If your diversity goes down in a given region, you have fewer polymorphic markers to get information from, and a sweep size affects a given limited region. So as your diversity goes down, your data points to detect that go down, your power goes down. It's kind of a broad way to think about it. If you're really worried about bottlenecks, do whole genome sequencing, because SNPs and singletons may give you some power to get around those we talked about. We'll talk about the singleton density score a little bit later on tomorrow. That'll be one way you can do to get around that. Yeah? Sure. So actually, spatial models have a long history. So Fisher and Kolmogorov had Fisher's traveling wave, which is an adaptive allele sweeping through a two-dimensional landscape. And there are simulation models to handle more complicated scenarios. The issue is when people do simulations to look for what they expect under neutrality, they often assume a fixed boundary for the population, and then fiddle with population structure and fiddle with bottlenecks and expansion and contraction, but don't actually map a pixelated, expansion in that range, which will give you very different values. So it's something you, in theory, can do. It's not something you can do analytically because each situation is different, right? It depends upon the topography, but it's something you could pretty easily, do with simulations. You basically have a template. You then throw in the geography, which would have, migration bottlenecks and migration, expansions, and just run several different scenarios and sort of see what the results are. And the bottom line is you probably couldn't detect sweeps versus bottlenecks. Now, again, there are other things you can look at. You can look at multiple factors and try to piece those together, as we'll talk about a little bit later on. I should mention, I'm probably overly pessimistic about stressing allelic surfing, but I think it's good for you to be cautious because people are so excitedly blind when they talk about detecting selection, because it's so fun and we all want to do it, right?